Yeah, morning someone told me, Vasista.
Yes, sir.
Yeah, can you summarize what you have understood from the morning class?
Sir, I wasn't there in the morning class.
You are not there,
No, sir.
but you said you were present.
So I was talking about Thursday, sir.
Okay, then that means, okay, I will change attendance.
Sir, but now,
Next, roll number Harjit.
Yes, sir.
Yeah,
So we have been talking about polysaccharides, homopolysaccharides,
yeah,
heteropolysaccharides. Homopolysaccharides contain structural and storage. Heteropolysaccharides contain extracellular components.
heteropolysaccharides.
Heteropolysaccharides contain extracellular components like mucopolysaccharides and all.
Ah,
Then we have been talking about glycogen, which it stores storage of polysaccharides. It's also
yeah.
alpha 1 to 4 and alpha 1 to 6 and cellulose.
So
Yes,
how can you say that alpha 124 126? What is this? You
polymer
you have
glycose.
already studied this course previously, and it is only the repetition for you, isn't it?
Yes, sir.
How can you say alpha 124 and alpha 126? What is this? 124. There are three numbers, huh?
No, so it's 1 to 1.
I think you please keep your mic in mute. I think I will only summarize. So in the today's morning class, we have discussed about the polysaccharides, and we said initially that these polysaccharides are long polymers, and several monomeric units are connected by the glycosidic bond. And these polysaccharides also called as glycans, and they have the high molecular weight and low molecular weight polysaccharides are water soluble, whereas the high molecular weight polysaccharides are water insoluble. And these polysaccharides are classified into two categories. And one is homopolysaccharides, second one is heteropolysaccharides. And these homopolysaccharides are made up of with only one type of sugar residues, sugar units, whereas the heteropolysaccharides are made up of with more than one type of sugar residues. And coming to the homopolysaccharides, we said that these homopolysaccharides have either structural role or the storage function. And for example, cellulose and the chitin, they have the structural role, whereas the starch and the glycogen has the storage function. Coming to the heteropolysaccharides, we said that these heteropolysaccharides are present in the extracellular space, that is the space between the two cells. And these heteropolysaccharides, they present in the extracellular space, have various functions. In the sense, they can hold the cells together and also provides the shape and support to the cells and tissues when they are present in the extracellular space. Then we discussed about initially the homopolysaccharides, the starch, cellulose, and glycogen. And in addition to this, we also discussed about the dextran, inulin, and other. And we said that initially we discussed about the starch, and it is a polymer of glucose. and it has the two components, amylose and the amylopectin. Amylose is a polymer of glucose with alpha-1 for glycosidic linkages. Amylopectin is a polymer of glucose with alpha-1 for as well as alpha-1,6 linkages. And the alpha-1,6 linkage is present only at the branch point, and this branching occurs for every 20 to 30 residues once in the chain. And this is this is, as I already said, that it is a the storage polysaccharide in and also reserve food material in the plants. Coming to the glycogen, the structure of the glycogen is similar to the structure of the amylopectin component of the starch, and the only difference is that the glycogen also has is a polymer of glucose with alpha-1,4 and alpha-1,6 glycosidic bonds, but the branch points are more in case of glycogen compared to the amylopectin component of the starch. The branch points occurs for every 5 to 6 residues once in the structure. And in the human body, the excess of glucose is stored in the form of the glycogen, mainly in the liver, muscle, and also in other tissues. And this so when the body demands energy, and again this glycogen is again undergoes degradation by glycogen phosphorylase enzyme, and as a result that glucose is utilized for the energy. And so in the human body, the stores the excess of glucose in the form of glycogen, and in order to prevent the generalized damage caused by the glucose if it is stored in its monomeric form, and which in turn drastically raises the cell osmolarity. So that means if the glucose is stored in its monomeric form, the osmolarity of the cell drastically increases. As a result, the cell death takes place. So that's why the glucose is stored in the human body, not in its monomeric form in the circulation, but it is stored in the in the form of the glycogen in the cells. And then we discussed about the structure of the cellulose, and it is a polymer of the glucose, again with beta-1,4 glycosidic bonds, and it is this cellulose is mainly present in the cell wall of the plants, and you can see here the cell wall is composed of several cellulose microfibrils, and each microfibril has linear chains of cellulose, and all these linear chains are associated with each other by the intermolecular hydrogen bonding, and this intermolecular hydrogen bonding gives rigidity to the cellulose. And since it is a polymer of glucose with beta-1,4 glycosidic linkages, most of the animals cannot use cellulose as a fuel source because they lack the enzyme to hydrolyze the beta-1. Ruminant animals and also the termites can digest the cellulose because they have the certain microorganisms in their gut. They can secrete the cellulose enzyme. And then we discussed about the dextran, and it is a polymer of the glucose with alpha-1,6 linkages, and with a branch point at alpha-1,3 and in some cases alpha-1,4. And this is this dextran is also the polysaccharide present in certain bacteria and also the yeast, and the dental plaques are rich in the dextran, and it is because of the certain organisms present on the teeth to secrete the dextran. And then we discussed about the synthetic dextran, that is Cepadex, is used as a stationary phase material in the gel filtration chromatography for the separation of molecules based on the size. And then we discussed about the chitin, and it is present in the exoskeleton of many arthropods such as insects, lobsters, and crabs. And chitin is a polymer of N-acetyl glucosamine with beta-1,4 glycosidic bonds. And of course, there are different types of chitin parallel and the anti-parallel chitin. This I have not highlighted here. Then inulin, and inulin, it is a storage form of the carbohydrates in tubers, and it is a polymer of fructose with terminal glucose unit, and the linkage here between the fructose units is that the beta-1 to 2,1 linkage, and these inulins are mainly the storage form of the polysaccharide, especially in some tubers like dahlia, artichoke, and garlic, and so on. Then we discussed about the some of the heteropolysaccharides, mainly peptidoglycan, and it is multi-layered in gram positive, and it is very thin in the gram negative, and chemically it is composed of the N-acetyl glucosamine and N-acetyl aromatic acid with the beta-1,4 glycosidic bonds, and these one NAG and NAM layer is cross-linked with another NAG and NAM layer through the short peptides, and this short peptide linkage is catalyzed by the transpeptidase enzyme, and the lysozyme enzyme hydrolyzes hydrolyzes the glycosidic bond present between the NAM and NAG. As a result, the organism, the lysozyme kills the bacteria, and in the same way, the penicillin also kills the bacteria by inhibiting the transpeptidase enzyme, which is essential for the formation of the peptide bond between the linear chains of the NAG and NAM. Then we discussed about the agarose, and it is present in certain marine red algae cell walls, and it is a sulfated heteropolysaccharide, and it is made up of with the D-galactose and 3,6-anhydro L-galactose, and it is also used as a as a supporting matrix for the agarose gel electrophoresis. We have already said that for the separation of the DNA. And then we discussed about the glycosaminoglycans, and there we have highlighted the glycosaminoglycans are also known as GAGs, and these are complex sugars, and we have already said that these glycosaminoglycans are mainly present in the extracellular space, that is the space between the cells. You can see here, and we have already said that they have many functions in the extracellular space because they are known to interact with many proteins such as chemokines, cytokines, growth factor, morphogens, enzymes, and so on. Then these glycosaminoglycans, when they are present in the when in aqueous solution, these glycosaminoglycans can hold large volumes of water. So that's why when there is a compression, we have already showed some picture. I think it is in the previous. They hold large volumes of the water, and then they have the various functions in the cells, and coming to its structure, as I already said that these glycosaminoglycans are first classified into two categories. One is non-sulfated GAGs, second one is the sulfated GAGs. In the non-sulfated GAGs, we have only hyaluronic acid, whereas the sulfated GAGs, we have only the keratan sulfate, the chondroitin sulfate, and the dermatan sulfate, and so on. And whether it is the sulfated GAG or the non-sulfated GAG, all these glycosaminoglycans are the polymers made up of with several repeating disaccharide units, and each disaccharide unit has one uronic acid residue, one amino sugar, and the uronic acid is either glucuronic acid or L-udrinic acid. The amino sugars are glucosamine or the galactosamine. I think that's where we have explained. Now coming to we will explain all these the glycosaminoglycans one by one in detail. So first one is the hyaluronic acid, and this hyaluronic acid is present in all living organisms, and it is and chemically it is a polysaccharide that is made up of with several disaccharide repeats of D-glucuronic acid, and N-acetyl D-glucosamine. So you can see here that it is a glucuronic acid, N-acetyl glucosamine with beta-1,3 and beta-1,4 alternate linkages. So that means throughout the glucuronic acid, N-acetyl glucosamine, glucuronic acid, N-acetyl glucosamine, it is the polymer of several disaccharide repeats consist of the glucuronic acid and N-acetyl glucosamine with the beta-1,4 and the beta-1,3 alternate linkages. And this hyaluronic acid is a very the high molecular weight polymer, and its molecular weight is up to 6,000 to 8,000 daltons. And this it is mainly present in the extracellular matrix and the pericellular matrix, and also seen in the very smaller amount in the intracellularly. And in Greek, halos means transparent. That means it has the translucent appearance, and it is mainly present in the skin and the vitreous of the eye. You can see here, this is the vitreous. So what you are seeing, the gel-like substance here, and vitreous of the eye and umbilical cord, and also the synovial fluid. So this hyaluronic acid is also present in the synovial fluid, and synovial fluid, we know that it is the fluid that is present in the synovial cavity. And in addition to this, it has many functions in the biological functions, in the sense since the hyaluronic acid can hold large volumes of the water, it is highly hygroscopic molecule, and it provides the viscoelasticity, or as a result, when it is present in the synovial fluid of the joints, it gives the lubrication to the joints, and it also regulates the tissue dynamic, the water transport, and also the tissue hydrodynamics, and the cell detachment, mitosis, and the tumor development and the metastasis, and so on. And as I already said, that the major function of the hyaluronic acid is only to bind the large volume of water, and as a result, it provides the lubrication to the mobile parts, especially the joints and in the muscles. So therefore, it is used also as a excellent the therapeutic the dietary supplement in the dietary supplement for treating the osteoarthritis and so on. And it is also, you can see here, it is also used in various you can see here that it is not only used as a therapeutic dietary supplement, it is also not only used as dietary supplement, it is also used as used in the night creams, in the sense these when it is present in the creams, that provides the hydration to the skin. And it is also used as the injections for as an anti wrinkling agents, and it is also used as a injectable form for treating the osteoarthritis and so on. So that means the hyaluronic acid has when it is usually normally it is present in the synovial fluid, and also in the joints, where the synovial cavity, where what you are seeing here is the synovial cavity. And when there is a the less amount of the synovial hyaluronic acid, then it causes the wear and tear in the joint. So that's why the hyaluronic acid is also used as a dietary supplement and as the injectable form of the agent for the treating the osteoarthritis. And of course, in the human body, this hyaluronic acid is degraded by enzymes known as the hyaluronidases, and these so the synthesis is taken place by the hyaluronic acid synthesis, whereas the degradation is mediated by the hyaluronidases. And in the body, or there are two types of receptors present for the hyaluronic acid. One is the CD44, second one is the receptor for hyaluronic acid mediated mobility. And these receptor concentration as well as the hyaluronic acid concentration is very low in the normal cells, and it is overexpressed in the many cancerous tissues, and which in turn promote the tumor metastasis. And then this hyaluronic acid is also used as it has many other biological functions, and It is also used as a drug vehicle agents because, as I already said, that this hyaluronic acid is composed of glucuronic acid and glucosamine with beta 1,4 and beta 1,3 alternate linkages. It has almost 50,000 repeats of this disaccharide. That means it is a very, very lengthy molecule, and so it has many other functions as well. It is also used as the drug vehicle agent, the vehicle agent, and of course it has many other properties as well. So this is about the hyaluronic acid, and in summary, the hyaluronic acid in Greek, halos means transparent, and it is mainly present in the synovial cavity. You can see here, and also the vitreous of the vitreous fluid of the eye, and it is a highly hygroscopic molecule, and it can hold large volumes of water, and its mainly major function is only to lubricate the mobile parts such as joints and the muscles. And in addition to this, this hyaluronic acid is also involved in the response, involved in the receptor mediated the cell detachment and the mitosis, the migration as well as the tumor development, and it is also used as the drug vehicle agents. In addition to this, and this hyaluronic acid is also used as a surgical aid in the ophthalmology in order during eye surgery, because in order to maintain the hydration in the eye. So this is about the functions of the hyaluronic acid. Now coming to the chondroitin sulfate. So chondroitin sulfate in Greek, chondros means the cartilage. Then that means it is mainly present in the cartilage and also the tendons and the ligaments and walls of the aorta. And this chondroitin sulfate, chemically, you can see here that it is composed of glucuronic acid, N-acetyl galactosamine 4 sulfate. So this is the, you can see here, this is the 1, 2, 3, 4, fourth carbon. So this is the glucuronic acid, and this is the N-acetyl galactosamine 4 sulfate, and with beta 1,3 and beta 1,4 alternate linkages. And this chondroitin sulfate is also present in the cornea and the retina of the eye. And as the age increases, the chondroitin sulfate concentration also decreases in the cartilage, and so that's why this chondroitin sulfate is also used as the supplement for treating osteoarthritis, where the wear and tear is present, because the chondroitin sulfate is mainly present in the cartilage, and it provides the tensile strength to the cartilage, tendons, and the ligaments. And this chondroitin sulfate is also used as the supplement for treating the osteoarthritis, and there are different types of chondroitin sulfates present. Chondroitin sulfate A, B, C, D, E, and the chondroitin sulfate B is known as the dermaton sulfate. Here the glucuronic acid is replaced with the L-udrinic acid. As I already said that this is the L-udrinic acid as well as the N-acetyl glucosamine 4 sulfate. And this dermaton sulfate is similar to the chondroitin sulfate, but here the glucuronic acid is replaced with the L-udrinic acid, and this dermaton sulfate is also present in the skin, blood vessels, heart valves, tendons, and lungs, and so on. And it has many other functions like coagulation and the wound repair and so on. And then coming to the keratin sulfate, coming to the keratin sulfate. So in Greek, keras means horn. That means it is present in the horny structures. And this keratin sulfate is peculiar in the structure. You can see here that instead of glucuronic acid or udrinic acid, it has only the galactose. So there is no uronic acid in the case of the keratin sulfate, and it is a polymer of the galactose and N-acetyl glucosamine 6 sulfate with beta 1,3 and beta 1,4 and beta 1,3 alternate linkages. And this keratin sulfate does not have the uronic acid, and it is also present in the cartilage, bone, and the cornea, and it also present in the variety of horny structures like hair and hoop nails and so on. And this keratin sulfate is classified. There are two types of keratin sulfates present. One is the keratin sulfate 1, and the second one is the keratin sulfate 2. This keratin sulfate 1 is mainly responsible for the corneal transparency, whereas the keratin sulfate 2 is mainly responsible for is mainly present in the loose connective tissues. So the only difference between the keratin sulfate 1 and the keratin sulfate 2 is that the site of attachment of the keratin sulfate to the protein. Keratin sulfate type 1 keratin sulfate 1 attaches to the asparagine residue of the protein, whereas the keratin sulfate 2 attaches to the serine or the threonine residues of the protein in order to for the formation of the proteoglycans. And deficiency of the keratin sulfate also leads to the disease called macular corneal dystrophy. You can also see here MCDC, macular corneal dystrophy, and it is the in this the stroma of the cornea is affected, and the synthesis of the keratin sulfate is either is absent or the abnormal, and that leads to the macular corneal dystrophy, and it is the autosomal recessive condition. So this is about the keratin sulfate. In summary, the keratin sulfate is a polymer of the galactose and N-acetyl glucosamine 6 sulfate, and it is mainly present in the cartilage, bone, and the cornea, and also the variety of horny structures. And this keratin sulfate, there are two types of keratin sulfates present. One is keratin sulfate 1, second one is the keratin sulfate 2. Type 1 is mainly responsible for the corneal transparency, whereas the type 2 is mainly present in the loose connective tissue. The only difference between the KS1 and the KS2 is that there is site of attachment to the protein for the formation of the proteoglycans. Deficiency of the keratin sulfate or the abnormal formation of the keratin sulfate leads to a condition called the macular corneal dystrophy. And this is about the keratin sulfate. And last we have the that is the heparan sulfate. And the heparan sulfate, I think we the yeah. So this heparan sulfate, in Greek, hepar means liver. So that means it is produced by almost all animal cells, and it is chemically it is composed of glucuronic acid 2 sulfate. You can see here glucuronic acid 2 sulfate, and also glucosamine 3 sulfate, and also glucosamine 2 sulfate and 6 sulfate. And this heparin is the fractionated form of the heparan sulfate, and it is also used as a injectable anticoagulant. And this heparan is a highly negatively charged molecule, because you can see here it has many negative charges here, because glucuronic acid 2 sulfate and also glucosamine sulfate and 3 sulfate and the 6 sulfate. So that's why it is a highly negatively charged molecule. And this because of this negative charges, it interacts with the many molecules present in the extracellular matrix as well. So this is about the glycosaminoglycans. In summary, these glycosaminoglycans are classified into two types: the non-sulfated and the sulfated. Non-sulfated glycosaminoglycan is only the hyaluronic acid. Sulfated glycosaminoglycans are chondroitin sulfate, dermatan sulfate, heparan sulfate, and the keratin sulfate. And hyaluronic acid is composed of is a polymer of the gluconic acid and N-acetyl glucosamine disaccharide repeats, and this disaccharide repeat present up to 50,000 times such. That means it is a very lengthy molecule. And coming to the chondroitin sulfate, and this chondroitin sulfate is again is a polymer of disaccharide repeat composed of the glucuronic acid and N-acetyl glucosamine 4-sulfate, and it has this disaccharide repeat. This disaccharide repeat is present from 20 to 60 times in this structure. And the dermaton sulfate, as I already said, that it is similar to the chondroitin sulfate B, and where the glucuronic acid is replaced with the alloderonic acid, and then the keraton sulfate. And this keraton sulfate, as I already said, that it doesn't have the uronic acid. It is composed of the galactose and N-acetyl glucosamine 6-sulfate repeat, and this disaccharide repeat present up to 25 times. So then last one is the heparan sulfate, and this heparan sulfate is chemically made up of the disaccharide units, consists of the glucuronic acid 2-sulfate and glucosamine sulfate and 3-sulfate and the 6-sulfate, and it is used as a anticoagulant. And in summary, hyaluronic acid is mainly responsible for the lubrication of the joints, and it is also present in the vitreous of the vitreous humor of the eye. And as the age advances, the hyaluronic acid concentration also decreases, and its association, hyaluronic acid concentration decreases. That's why the most of the people in their with age, they suffer with the various vision problems. And in addition to this hyaluronic acid is also present in the synovial fluid, and that provides the lubrication to the lubrication. And then chondroitin sulfate is mainly present in the cartilage, that gives the tensile strength to the cartilage. And dermaton sulfate mainly present in the skin and also the other this one. Heparan sulfate, the heparin is the fractionated form of the heparan sulfate. It is used as the injectable anticoagulant. Keraton sulfate is mainly responsible for the corneal transparency as well. So this is the view of the glycosaminoglycans. Now coming to we have the another topic in this, that is the proteoglycans. So coming to the proteoglycans, before understanding the proteoglycans, let us understand what are glycoconjugates. So we know, we have already said in the last class that these the glycans are the carbohydrate chains that are present on the cell surface, have various functions. As we have already said, that what we are seeing here is the glycan chains. They are involved in the cell-cell interaction, cell-cell recognition, and cell-cell adhesion, and so on. You can see here that the carbohydrates that are present on the surface of the cell membrane involved in various recognition, molecular recognition processes, such as the interaction of the virus to the host cell or the hormone to the cell, and many. So all these are because of the glycan chains that are present on the cell surface. So now these glycans either present on the cell surface, either they are attached to the protein, or they are attached to the lipid. So that means if the carbohydrates, when they are covalently linked to the protein, then the glycoproteins are formed, and when the carbohydrates, when they are linked to the lipid, and the glycolipids are formed. So that means these are glycoconjugates. And glycoconjugates means the carbohydrates that are linked to the non-carbohydrate moieties. Here, in case of glycoproteins, the carbohydrates the linked to the proteins, whereas in case of glycolipids, the carbohydrates are linked to the lipid in glycolipid. And in both the cases, in both the glycoproteins as well as the glycolipid, the major component is from the protein and as well as the lipid. But the major component in case of glycoproteins, the major component is contributed by the protein, and the minor component is from the sugar. And in the same way, but in the glycolipid also same. But in case of, we have the another third class of the glycoconjugates. Those are known as the proteoglycans. So in the proteoglycans, the glycosaminoglycans are covalently linked to the proteins. So they are known as the proteoglycans. And in this, the major component of the proteoglycan is contributed by the gap chains, and the minor component is the protein. That is only the difference between the proteoglycan and the glycoproteins. You can see here in the proteoglycan are the glycoproteins. In glycoprotein also composed of the carbohydrate as well as the protein. Proteoglycan is also composed of the GAGs and the proteins, but here in case of proteoglycans, the major component is the major portion is contributed by the GAGs, minor portion is from the protein, whereas in case of glycoprotein, major portion is from the protein or the minor portion is from the carbohydrate. So that is so all these three are known as the glycoconjugates, where the carbohydrates are linked to the non-carbohydrate moieties. And so we discuss about in today's class we discuss about the proteoglycans, and these proteoglycans are the glycosaminoglycans that are covalently linked to the proteins. Those are known as the proteoglycans. And in proteoglycans, you can see here that these glycosaminoglycans are covalently linked to the core protein through a tri or the tetrasaccharide bridge, and the site of attachment is at the serine residue. That means the proteoglycans are composed of a core protein, and several gap chains are covalently attached to the core protein through tri or the tetrasaccharide bridge, and the site of attachment is the serine residue. And the serine residue should be present in a particular order, that is the serine, glycine, X glycine. So the site of attachment is the serine residue, and the serine should be present in a particular sequence, that is the serine and glycine, X glycine. Here X can be any amino acid, but the next to the serine should be glycine. Then only the serine could be a potential site for the GAG attachment. If any bulky amino acid is present at the near to the serine, then this cannot act as a potential site for the GAG attachment. And these proteoglycans are further classified based on the type of the GAG chains that are covalently linked to the protein. They are again classified into the chondroitin sulfate proteoglycans, heparan sulfate proteoglycans, dermatan sulfate proteoglycans, and so on, but not the heparan, the hyaluronic acid proteoglycans, because hyaluronic acid does not form the proteoglycans, but it in turn forms the various supramolecular assemblies with these proteoglycans, which in turn forms the various supramolecular assemblies with the hyaluronic acid. This we will see later. And so, what is the time? Yeah. We will continue this topic in next class. I think I will record this and put it in the MS team. You need not attend to that class. I will record tomorrow. Tomorrow, do we have class, students? Hello. Tomorrow, do we have class or day after tomorrow?
Day after. Tomorrow we don't have class. Day after tomorrow, sir.
Okay, if it's I will try to complete this one, and tomorrow you need not join the meeting. I will schedule the meeting and I will record this lecture. You can go through the lecture such that we will try to complete this module.